lymphoma. If you used Roundup week.
Okay, good morning, everyone. It's great to be here to share some of our current research with you and to talk about the epidemiology and spread of Lyme disease in built environments, a problem that is growing and now affects, of course, 14 states in the Northeast and upper Midwest, where about a quarter of the U.S. population lives. There are other states that have, many others that have Lyme cases, but the foci are in the upper Midwest and, of course, the Northeast. Well, the emergence and spread of Lyme, some of you are, of course, have seen these maps and are aware of the spatial distribution and scope of the growing problem of Lyme disease in the U.S. These CDC maps that I'm going to show you represent cases from 2001 to 2017 and highlight the problem. And they represent an exponential growth in Lyme disease incidence. And if you can just watch and see as we course through these, you'll see the significance of the problem. Now, Lyme disease is certainly not declining in New York, and it's certainly exploding to our south in Pennsylvania. And it reaches an incidence per 100,000 population in Pennsylvania that we see in New York. And very recently, as you can see in this graph, it surpasses New York. And part of the increase, of course, is due to the recognition and reporting by the medical community, as well as increases in the tick vector and, of course, the reservoir host for Lyme, for the Lyme bacterium, which I'll get into in just a minute. If we look at the Lyme incidence in New York, the rate per 100,000 population, these are figures from the New York State Department of Health. I've highlighted, of course, two regions, one that we're working in and the other that many of you may live in. And you can see that the crude rate for the southern tier is approximately 110 cases per 100,000 population. And Western New York in yellow is relatively low by comparison, of around 4.5 cases per 100,000 population. And that's reported by region, but I also want to point out that there is county variability. If we look at the southern tier region, Delaware County has about 92 cases per 100,000, Tioga 127, and leads the southern tier region, but different from Western New York, at least according to the latest figures. Erie County has 4 cases, and Cattaraugus about 12. So you can see a threefold difference just here in Western New York. Why the difference? Well, part of it, of course, deals with case ascertainment. Certainly a significant portion, in my opinion, underreporting of cases of Lyme, as White and colleagues recently demonstrated. Other reasons, of course, include the spatial distribution of the vector. The black-legged tick or deer tick, Ixodes scapularis, from county to county, and of course within counties. Nothing is uniform, okay? There's this geospatial variability that I'll be talking about a little bit more in the competent reservoir for the Lyme bacterium, the white-footed mouse that Jill showed you earlier, Paramiscus leucopus, and I'll go into detail a little bit more on the vector and the reservoir host in just a moment. And we're going to cover a little bit about ticks, the life cycle stages, the reservoir, and the infectious agent. Of course, as all of you, I'm sure, are aware, the causative agent of Lyme is the bacterium Borrelia burgdorferi, and other subspecies discovered 1981. Willie Burgdorfer, the Rocky Mountain Labs. It's tightly coiled, spirochete. It's very fast moving, and as you'll hear probably throughout the day, can evade the immune system. One of the comparisons, Jill showed you a little bit earlier, the relative comparison of the size of the ticks. We're looking at the various life cycle stages. Larvae, by the way, one way to tell is that they have 6 legs, whereas the nymphal and adult ticks have 8. So if you're unsure of recognizing, you can just count the legs, and if it has 6, it's larval stage. The larvae are really tiny, and with my advancing age, I can't see it with the naked eye, one single larval tick. I can see nymphal ticks yet, and of course adults, but there's a great difference in size between them. And the larvae are also lighter colored, which makes it more difficult to see as well. Well, this is the reservoir host, the primary competent reservoir host. What do I mean by competency? For the Lyme bacterium, that is about 90% of the time when this little guy is infected, it'll pass that infection onto the tick that feeds on it, usually the larval stage tick, because they're so small and the mice can't groom them off very easily. Okay, so that's when the larvae, who are uninfected, when they hatch out from the egg stage, become infected when they feed and take a blood meal. They can, on this little guy, and 90% of the time when they do so, they become infected. That's why this white-footed mouse is so important as a reservoir. Well, transmission happens, of course, during feeding. The Lyme bacteria live in the gut of the tick. They're triggered to move when the tick starts to feed, when it starts ingesting blood. So the bacteria migrate out of the gut into the saliva, and then they are inoculated into a person or one warm-blooded animal that feeds opportunistically on the mouse. The ticks feed pretty slowly in the beginning, so the process of infection takes a while. The general rule of thumb that CDC puts out is 36 to 48 hours after being attached. That transmission usually proceeds, but actual transmission of the infectious agent can take place in less than 24 hours. And that's why it's so important to check for ticks and not wait. Okay, so it can happen, and I think experimentally it's happened even under experimental conditions, even under 6 hours. So you have to pay attention and get and have those tick checks as soon as possible. Now, a little about co-infections. I don't want to spend a lot of time on this, but the black-legged or deer tick carry, of course, as Jill mentioned, several human infectious pathogens, including not only Lyme, but Anaplasma, Babesia, Powassan virus. This particular tick, the black-legged or deer tick, and they, of course, have some symptoms similar to Lyme in case of Anaplasma. Babesia is intracellular, red cell, somewhat similar to malaria. And there are other pathogens and other ticks, as Jill indicated, that produce disease. You have the Lone Star tick, you have the dog tick, etc., that all produce human infections or carry human infections. I want to now move on to, with this background, a little bit update about our research, which involves an understanding of the risk of Lyme and fragmented landscapes, okay, within built environments. And our goal is to determine the prevalence of Lyme pathogen in non-contiguous, physically fragmented spaces within built environments, and the development of predictive risk models of transmission of Lyme and other tick-borne illnesses. Okay, and by physically fragmented spaces, we refer to spaces that are physically broken up by man-made structures. So we're truly talking about urban, peri-urban areas, or any areas that are broken up by physical man-made structures with green or ecological, small fragmented ecological spaces in between. So if I were to show you this photo and ask you where you thought the highest incidence of the infectious agent would be found, where would you think? Here, okay? So these are the areas that we're working in. And we define built environments according to Srinivasan and colleagues, where human-modified places such as homes, schools, workplaces, parks, industrial areas, roads, highways, all of these built environmental structures, and neighborhood backyards that serve as humans' most important and most frequently used habitat. Okay, so this is an important means for understanding the risk of Lyme disease, which we believe can then be used for public health mitigation strategies. One of the aspects of fragmented landscapes has to do with a comparison of the intact forests, and Jill mentioned the Carey Institute and Rick Osfeld. In an intact, heavily healthy forest, biodiversity is high, and the white-footed mouse and other rodent populations are kept in check by predators, which in turn keep the tick populations low. If we look at fragmented forests broken up by man-made structures, biodiversity decreases, and there are really few predators like fox and hawks. And thus the mouse and other rodent populations increase, and in turn both the tick and Lyme disease thrive. So if we put this into sort of a hypothesis form relative to our research efforts, our central hypothesis is that fragmented ecosystems within built environments that have high human foot traffic or high human activity create areas where contact with the infectious agent for Lyme is extraordinarily high. I don't have time to go into all of the details of what humans do to cause that interaction and thus risk to increase, but I'll mention a few things a little bit later on. So you have to have obviously infected ticks, you have to have the infectious agent, the infected ticks, you have to have a reservoir host, and then you have to have the secondary host, which is humans or domestic pets, okay, primarily. And you also associated the population of ticks are associated with environmental variables such as rainfall, temperature, forest cover, as we mentioned, and all of these things together determine the geospatial difference in tick populations, where it might be high or low. And that's why I mentioned early on why you might be seeing in part some fairly different figures, in addition to underreporting, very different figures in terms of Lyme cases. So this is kind of what the kind of model that we're building, but there's yet another layer. We've been talking about environmental factors, of course, but we have an additional interaction, and that's human social, behavioral, and demographic elements that you have to overlie on all the ecological factors that I've been talking about that contribute to individuals encountering ticks. And I'll tell you a little bit more about that. And again, keep in mind that we're talking about built environments primarily. And I'll tell you a little bit about one of the projects, the first project, which we call the Parks Project. And it's a survey of some 16 parks in a 6-county area of the upper Susquehanna River Basin. We looked at tick populations. We trapped the white-footed mouse in high-traffic, high human activity areas. Again, where people are is where we're looking at risk for Lyme. So not out in a remote area of the park, but where the primary activity of people are, where the greatest demographics are in the park. And we look at density and prevalence of the infectious agent, and I'll tell you how in just a minute. This slide just shows you some of the 16 sites, 16 parks that we've been working in the 6-county area of the upper Susquehanna River Basin. We've got Tioga and Tompkins, Broome, Shenango, Otsego, and Delaware counties that make up that region. One of the things, of course, as I mentioned, how do we drag for ticks? We use a 1 meter by 1 meter drag cloth, as you see here, and trails in a park. We drag on either side of the walkways to collect the ticks. We trap for mice as well, just off of these walkways. As far as the ticks go, we collect them on the drag. This is a corduroy drag, and it's easy for the ticks to kind of hang on, and we're able to pick them off from the drag. And we just to give you an idea of density, the Binghamton University campus has a density of about 7.5 ticks per 1,000 square meters. And you can see variations again by county. So we compared the campus, which is about a 910-acre campus, and then the county parks in all of the state and county parks in the 6 counties themselves. And you can see the variation that we wind up having. What do we do with them? We take them and identify species, life cycle stage, sex. We extract DNA then from the ticks. We emulsify them using liquid nitrogen, and we amplify them molecularly, and we isolate the DNA of the spirochete. That's what we're searching for, okay? So it's the pathogen. We're not interested in really the tick DNA per se, in terms of trying to find out whether or not they have the, we're interested in the bacteria that they carry. Okay, this gives you some idea of the prevalence the percent infected ticks. So you have total ticks, nymphal ticks, and adult ticks. You can see that in our area, this upper Susquehanna River Basin, you have some variation, but overall, the average of all infected ticks is running somewhere between 30 and 40+%. If you look at nymphal ticks, you'll see it's down to generally 25 to 30%. And if you look at the adult ticks, you'll see that the infection rate is twice as much as nymphal ticks. And this is because they've taken 2 previous blood meals, so they had 2 prior chances as an adult of becoming infected than at the nymphal stage, okay? So that's generally the difference. I think all told, we probably dragged some 193 miles in these state parks. So we had a pretty hefty program going. And the size of the, I think I have the density here. Well, the density of the infected nymphs themselves, a high-risk density would be approximately 0.3 infected nymphs per 1,000 square meters, and we have, you can see on that graph, I have the black line at the bottom. You can see that, well, you can see the line anyway, and the counties there, most of them are above that high-risk line. So that line represents high risk for Lyme disease. I mentioned to you the issue of the reservoir host. So we track the reservoir host using an approved protocol at Binghamton. We identify everything by GPS coordinates, where we place the traps, etc. And we place them along those same high traffic walkways and paths of high human use. We look at the tissue of the white-footed mouse, and if we were just to rely on the bacterium circulating in the blood of the mouse, we'd only find about 1% to 2% of the mice infected. When we begin to look at where the agent is sequestered in the mouse, if we go to the organs themselves, we find out it's upwards of 30–35% of these mice are infected. So if we're trying to understand transmission dynamics, you have to understand that what you find in the peripheral blood of these mice is not the pathogen load that they're actually carrying, the rate of infection, particularly rather than pathogen load, but whether or not they're infected. I want to move on to a second project that's associated with the first, and that is understanding risk in built environments. And it's a new initiative that we've recently started, and it's conducted initially in the Triple Cities area in the southern tier, in neighborhood backyards. And I have a little bit about it here. Pretty much what seeks to determine the density and infectivity of nymphal and adult ticks in urban neighborhoods. Okay? We find that we had something like, I guess, 152 households studied so far. We've dragged all of the low-lying vegetation. I don't know if I have a schematic in here, I've forgotten. But base of all trees, so we take the periphery of the yard, we drag all of the vegetation, we drag 5-meter transects of the lawn, and we drag the perimeter of the house. That's essentially what we do in these rear yards. We find that the ticks are highly infected, and this represents the percentages of the area that we drag. The average percentage of the area that we drag. Property means the periphery of the property. House is the periphery of the house. In the rear yard, lawn, these 5-meter transects of the lawn, and vegetation being understory vegetation that we find in the yard. So you can see in this case that the amount of vegetation in the yard is small. The periphery of the property is 44% of what we drag. Lawn is 37%. When we look at the distribution of the ticks, we find that most of it is in the vegetation and the periphery of the property. The number of ticks in the lawn is quite low. And actually, we would expect that ecologically. So, what are we hoping to do kind of moving forward with this project? We want to dramatically expand it because we think that in terms of assessing risk of infection, that this type of study in places where people spend the majority of their time is an important part to consider in terms of risk assessment for Lyme. And we hope to add to the project use of drones that will give us a geospatial arrangement, not only of a given yard, but of a neighborhood. That'll give us the connectivity of the green spaces, not only yard to yard, but if there's any other green space within the area that might be conducive to high-density tick populations. And we think this has quite a lot of potential. So what we're planning to do is to take these 6 counties where we've been working in parks and look at 3 cities per county. So we'd have about 18 of them total, 3 neighborhoods per city, so something like 54 neighborhoods, and 25 households on average per neighborhood. So we've got about 1,300 residences or so that we hope to expand the project to. So we'll have a lot of work, of course, and hopefully we can find the funding to carry this project out. I want to talk a little bit about prevention. Jill mentioned a few things regarding it. We find that overall awareness and what we call situational awareness is key. Once people are aware, risk of disease goes down. Our goal is to model risk so that we can effectively come up with some intervention and prevention strategies once we have a model that has been validated and then translated to county health departments so that they can institute the appropriate measures to reduce risk of Lyme. Some of you have seen this. You can get it on the internet. I can't remember. I think it was originally maybe Connecticut or Rhode Island that had this modification. There are things you can do in your yard and that will probably help to reduce risk. We'll be able to evaluate all of this when we do our on-the-ground analysis in these residences, and then the use of drones and the geospatial arrangement of all of the connecting properties around a given property, so we'll be able to layer all of these things. And the one key layer, of course, that I didn't mention yet is that we put humans in the middle of this. That is, who uses the backyard of the residence, how many people use it, when do they use it, what do they use it for, do they have pets, are the pets indoor pets, outdoor pets, indoor, outdoor, okay, do they roam or not roam outside of the yard, etc. All of this data and follow up on whether or not they found any ticks in their yard, etc., will give us a good picture of the behavioral and demographics associated with the ecological measures and the geospatial measures from the drone. So it's almost 3 layers that we're looking at to be able to develop these risk models and then the mitigation and prevention models, we hope, that will come out of that using what we call a system dynamics approach in computer simulations, and then doing what's called what-if scenarios once we validate our risk models. I want to talk just a bit about deer. What role do deer play? There's been a lot of speculation about the role of deer in transmission of Lyme. They act as spatial distributors of ticks. They're a large mammal. Many of you know that they have a large home range, relatively large relative to other smaller mammals. So they distribute ticks. They pick them up, they drop them off. Ticks don't move around. They need somebody to carry them. You know, they're in a 1 square meter area, and that's as far as they're generally going to go. So they need a carrier. And these guys are a good carrier over a fairly large landscape, okay? They also act as the primary breeding site for ticks, okay? This is the time of year right now. And those of you that hunt deer, particularly in areas where there are lots of ticks, you're going to see potentially something like this on the facial areas and ears of these ticks, or between the legs, not on the heavy coat. Okay? They're going to come to where there are small blood vessels from which they can take a blood meal. That's their goal. They need that blood meal to go on to the next stage of their life cycle. But, and we've published a paper on this, they tend, in our estimation, using indirect evidence, to clear the infectious agent, to clear Borrelia. So they don't pass on. Ticks don't bite them and become infected. Okay? Much as what's called the California fence lizard. All right? They tend to clear the infection, and that's a paper we published here about 2 years ago that's gotten quite a lot of attention. But they do act as the spatial distributors and as a major breeding site. So yes, they play very major roles in maintenance of these tick populations. Another study we've just finished up, really, which is our work with household pets. Dog study that we've looked at, as you can see here, we've looked at 578 bloods from a veterinary clinic. These are bloods from dogs that were brought in for all sorts of reasons, not just because the owners thought they might have contracted Lyme. And we looked at the blood, and we found that 34% of them are carrying the pathogen. Because in the lab, we search for the DNA of the pathogen. We can't tell you whether that bacterium is alive, that Borrelia is alive, but we can tell you whether it was there. And 34% of the dogs, out of a fairly large sample, have the pathogen in them. Okay? So one of the things that you need to consider is first, if you find any ticks on your pets, dogs or cats usually, or if you have horses, you know, you should begin thinking and being alerted to the potential for having a problem, first of all, in your yard, particularly if they don't go outside of your yard. Interestingly, we just finished a study that we submitted for publication, and we found that pet owners are at a higher risk of Lyme than non-pet owners. And most interesting, cat owners were probably at highest risk, even more so than dog owners. If you think about it, it probably makes some sense. Depends on whether your cat's a roamer, whether you let your cat outside. And if you do, that cat generally isn't going to stay in your yard, is going to be able to hunt, potentially, birds, mice, etc., and is likely going to come in contact with ticks. I've had cat owners tell me that they've had problems, and cat owners tend not to look for ticks as critically as dog owners, probably because it's difficult with some cats to groom them, to play with them, to hold them still long enough in order to look significantly for ticks and tick burdens. I just wanted to mention a couple of things before I finish up on the Tick-borne Disease Research Center at Binghamton University. It's a transdisciplinary center for Lyme and other tick-borne diseases. It's only a year old, and the center is made up of a couple of dozen faculty from many departments and schools within the university, schools and colleges within the university, and has a number of collaborating SUNY institutions and associated hospitals and medical centers. There's three pillars of the center. One, which I've concentrated on fairly heavily today, which is ecology, epidemiology, and risk modeling. The second is diagnostics and treatment, and we're committed with the School of Pharmacy to look for biomarkers of both acute and chronic Lyme and other tick-borne illnesses, but we're concentrating in those areas. And then the third pillar of the center is public health education and outreach. And that's formed a fairly significant part of our program. So one of the things that we're hoping to do, of course, is to, as I mentioned, the diagnostic side, optimizing Lyme disease treatment and management. We're also working with public health departments to assist with risk management and assessment. And we want to extend surveillance much more broadly. We think that's important, and I've talked about the Parks and the Neighborhood Project as part of that. We have some 20 plus projects underway right now by different faculty members, and we bring, we have adult education courses through Lyceum, seminars, conferences, of course, but one thing we also do is reach out to elementary and middle school kids, and we bring them to the center and talk to them about Lyme. We go to the schools and do the same, Lyme and other tick-borne illnesses, and there seems to be a fair amount of traction in our area of schools that are interested in participating and having us be there and talk to the kids and begin to familiarize them with the problem. Predictions, sometimes we're asked, what's happening, where are things going? Certainly it's here to stay in the foreseeable future. These things take a long time to develop, and they take a long time to ramp down. So it's not going to happen right away. So you're going to have to consider how you're going to deal with the consequences of Lyme, both in built as well as wilderness environments. Jill talked about new ticks in the region. Federal and state funding is critical for research. Research is not a quick and dirty kind of a thing. It takes money, it takes time for us to get a consensus on exactly what's happening. So it takes a lot of patience, but in time, the research will pan out to lead us in the right directions to ameliorate risk and hopefully treatment as well. Vaccines, probably Bob Jagir may or may not talk about some of that in his presentation, where we stand with it, etc. There are some European countries that are in various stages of it. We have just a little bit of advertising. Some of you, of course, are aware and have been at our annual Lyme disease conference at the university. It's first week in May every year, and it's going to be May 2nd this coming year, 2020. And we feel that it has helped a lot. Part of that is the students. We have a large cadre of undergraduate and graduate students that are working with various investigators at the Tick-borne Disease Research Center. Amanda Rome on the right has gone on to take a position as a research scientist, graduated now as a research scientist at Bassett Healthcare Research Institute, and will be continuing to work on Lyme. They want her to develop a program there, so New York State is fortunate to have another Lyme researcher working on the problem in the Upper Susquehanna River Basin. This is just part of our tick-borne disease team, and these are going to be the next generation of scientists and physicians. So they'll be around hopefully to help some of them with the growing problem. Thank you.